Are you
Yeah.
an Apple
Next.
person?
So we should be able to escape it somehow.
Well,
Yeah.
yeah. There we go. And then Tuesday.
Yep.
Come on. Do you want to go for
Yeah,
it?
I think this might be set up with a left click. So it's
And then you're numbered, so you're Nobel.
yes. That's the one.
Yes, and you're a minute to the good, so.
Okay, good.
Okay, we're going to continue with the theme of cancer's genetics, and our next presenter is Robert Nobel.
Thank you, and thanks for inviting me. So I'm going to continue on a similar theme that was very nicely introduced by the previous speaker. The question I'm interested in is what kind of evolutionary mode will we expect within tumors? So here's a recent review that discussed evidence for various different kinds of evolutionary mode. Punctuated evolution, linear, where we get lots of selective sweeps, a branching evolutionary process, which has been seen in a lot of multi-region sequencing, and neutral evolution, which has been proposed particularly in a model of colorectal cancer. And what you can see from these pictures are they're simply cartoons. We don't have a very good model for why we would expect one of these patterns in one tumor type compared to another. And what you've just seen is a very nice talk about trying to infer these patterns from data. What I'm going to present today is a way of a complementary approach, which is doing forwards in time models to try and see why we would expect these different patterns to emerge. So what I've developed over the last few years is a model of intra-tumor population genetics. This is a stochastic model, although we can do some analytical analyses of it. It's an agent-based model where we represent cells and their evolution. And something that I've spent a lot of time on is making sure that this single modeling framework can represent lots of different kinds of spatial structure to allow us to compare different tumor types. Something unusual about this model, we can allow tumors to grow into empty spaces inside a duct, or we can model the invasion of tissue. And we can evolve the cell division rate or dispersal rate. So we have the acquisition of both lots of passenger mutations and driver mutations. A difficulty in these models is always parameterizing. So to do essentially the best we can with the data available, I teamed up with an expert in histopathological image analysis, Jakob Katter, who was able to take slides from the Cancer Genome Atlas and to quantify some aspects of the spatial structure of particular tumor types, and to apply his expertise as an oncologist to give us some inferred parameter values for the spatial structure of these tumors to set up the model. And what Jakob and I ended up with was 4 particular kinds of spatial structure that we would particularly examine. The first kind is leukemia, blood cancers, which is generally assumed to be essentially a non-spatial system, whereby cells are more or less well mixed with one another. The second example is precursors of colorectal tumors, particularly polyps, adenomas. These tumors, to some extent, recapitulate the structure of the healthy gut, in which we have glands of cells that divide in half. The third case, a lot of solid tumors show a glandular or acinar structure, where we have little glands of cells separated from one another. And these tumors grow by invading normal tissue. So we can represent that in the model as well. And the final case is boundary growth. This is just a ball of cells that expands. This model has been widely used for modeling tumors. It might be particularly appropriate for both benign tumors and perhaps liver cancers, it's been argued. So we take these 4 types, and what I wanted to do was to compare how the structure of the tumor, the architecture, and the mode of cell dispersal influences the evolutionary mode, putting aside the evolutionary parameters. So what I did was I ran many simulations in which I kept evolutionary parameters fixed, so in particular the mutation rate and the distribution of fitness effects of these driver mutations. And I just varied the spatial structure among the 4 paradigms that I've just shown you. And I vary whether we're invading normal tissue or growing into a duct. And here are the results in brief. So I do have a poster and we'll be happy to take people through in more detail. I also have a preprint so you can read about it. But very briefly, what we're doing on the left here is we're starting with the histopathology and other information about these different tumor types. We turn this into a parameterization of the model. We run this through the model. And then on the right, we see results. These are results in each row. This represents a single simulation, just represented in different ways. So the first panel there shows what's called a Muller plot. This is different clones which have different combinations of driver mutations represented by different colors and how their frequencies change over time. The second representation is the spatial layout of clones at the end, except in the top case, which is non-spatial, so that's just the distribution. And the final case is what happens if you draw a phylogenetic tree based on these driver clones. And what you see there is you get very different patterns. You get different tumor evolutionary modes just depending on the spatial structure of the tumor. You can keep the mutation rate the same, the distribution of fitness effects, you can keep everything else the same. Just the way that cells interact with each other gives you these different patterns. I want you to particularly look at the third row there, which is this branching evolution, this deep branching. And what's interesting about that is it's been reported in many studies of multi-region sequencing studies of different solid tumor types. In particular, there's a very nice recent survey by the TracerX Renal Consortium. In this case, they looked at more than 100 kidney tumors. They did multi-region sequencing, average of about 8 samples per tumor, and they reconstructed trees, and as you can see perhaps in that graphical abstract, in many cases they found these kind of branching patterns. So what I did was I took the data that was publicly available for a few of these tumors, and I summarized these trees with a couple of evolutionary metrics, and I compared that to my model results, and I find that indeed the model is fairly consistent. So on the left there you see the results of a lot of simulations, and the black points that are labeled, these correspond to particular tumors from the TracerX renal cohort. So this isn't meant to prove that my model is correct, it's just meant to show that it produces results in the right ballpark. It's kind of a sanity check at this point. So there are a lot more results I've got from this model, which I don't have quite time to talk about now, but the main conclusions I want to take you with you is that tumor evolutionary mode is plausibly governed by the kind of spatial structure within tumor types. This provides a simple mechanistic explanation based on something we can actually see, something that pathologists are aware of, rather than trying to invoke other mechanisms that are less well characterized. I think this motivates us to look more carefully at the parameters of cell dispersal and how these cells are interacting with one another. And it has obvious implications for how we go about forecasting tumor evolution and controlling it. So I'd just like to thank the people on the left who are my co-authors and the people on the right who've helped a lot as well. There's a preprint, there's code that's publicly available, and there's a poster I'll be presenting this evening. Thank you.
So do we have questions for Rob?
Very nice talk. So could you maybe comment, one thing you didn't allude to in this talk, didn't mention talk, is the different fitness landscape that tumors face.
Yep.
So each tumor faces a different, fitness constraint. Could you maybe say something about that?
Yeah, so I mean, this being an agent-based model, there are all kinds of fitness landscapes I could put into it very easily. For this first paper and this first set of results, I've assumed a very simple fitness landscape, which is simply multiplicative fitness. Each driver gives you a fitness advantage, and I have very weakly diminishing returns on that. But it's very easy to put epistasis and all kinds of different fitness landscapes into this model. I just think that that's a follow-up study in its own right, so I haven't looked into that yet.
Okay, thanks.
Do we have additional questions? I have a question, which is in the experimental data. How difficult is it to estimate the number of cells that have a particular driver mutation, and how does that affect your model?
Yeah, so the big limitation on this modeling, I think, is the data that's available. So the TracerX renal, what they did there was multi-region bulk sequencing. And then they reconstructed trees based on that. And there are all kinds of caveats about, we don't get very good estimates of the frequency of different types. There are other kinds of data, as you've seen, single-cell data, there's organoid data. But I think that what we're trying to do is develop the data and the models side by side, and eventually they'll converge. But at the moment, we don't quite have the data to really test these models.
Thank you. Let's thank Rob again.